I've created a small model to track generational changes in a small population. The model keeps track of who is whose parent and which genes were passed down. Based on this model, we'll explore: who are mitochondrial Eve and Y-chromosomal Adam? Where do they come from, and what have scientists proven? What other types of common ancestors exist? How closely related are all humans, and what is the identical ancestor's point? And also, what we have actually inherited from our ancestors. Let's dive into how the model we'll be using today works. Suppose we start with a population of 3 women and 3 men. We randomly distribute the number of offspring among the women so that 1/3 have 1 child, another 1/3 have 2 children, and the final 1/3 have 3 children. On average, each woman has 2 offspring, which keeps the population size stable. We then randomly assign a father for each offspring. This means some men may not have any offspring, while others might become the father of all the children in the new generation. In other words, men have a greater variance in reproductive success than women, which aligns with reality. We randomly assign the sex of the offspring to maintain an equal male-to-female ratio and copy the corresponding DNA segments from their parents. We place them in a table, with women listed first, followed by men. We position them, where possible, opposite their same-sex ancestor to make the diagram more visually clear. This setup allows us to trace ancestry through both the maternal and paternal lines. Here's a quick rundown of what we inherit from our parents. The human genome consists of 23 pairs of chromosomes. Each pair is made up of homologous chromosomes, one from the father and one from the mother. They contain the same set of genes, but the genes themselves might have slight differences. Your sex is determined by the last pair of chromosomes. Women have 2 X chromosomes, while men have 1 X chromosome and a smaller Y chromosome. Before a sex cell is created, a process called crossing over occurs. This is when pairs of homologous chromosomes exchange random segments of their genome. Since the last pair of chromosomes in males is not homologous, crossing over does not occur. In a woman's egg cell, 1 chromosome from each pair ends up, creating a single set of 23 chromosomes. In each chromosome, the genes from her father and mother are randomly mixed. This means the child receives half of their genome from their mother, but within that half, there will be a random proportion of genes from their grandmother and grandfather. The same thing happens with a man's sperm cell. It also gets a single set of chromosomes. If the sperm contains an X chromosome, a girl is born. This X chromosome, which avoided crossing over this time, will undergo it in the next generation. If the sperm contains a Y chromosome, a boy is born. The Y chromosome is passed down unchanged from father to son, and so on. This makes the Y chromosome a perfect tool for tracking a person's paternal lineage. Every organism begins its development from a single egg cell, and the egg cell is from the mother. In addition to nuclear DNA, our cells also contain mitochondria, which have their own small, circular DNA molecule. Since the egg cell is from the mother, all of her children receive mitochondria from her. Only women can pass these mitochondria on to the next generation. This makes mitochondrial DNA an ideal tool for tracking a person's maternal lineage. It's easy to show that all paternal and maternal lineages converge to a single man and a single woman, which I will try to demonstrate in this video. They are often called Y-chromosomal Adam and mitochondrial Eve. For brevity, I'll sometimes just refer to them as Adam and Eve. Before we run the model, it's important to grasp one key concept that holds significant importance in evolutionary biology. If a drunk person is walking along a road, with each step equally likely to veer to the right or the left, sooner or later, they will fall into a ditch. Knowing their starting position, you can accurately determine the probability of them reaching one side of the road or the other. If one side is absent, they will inevitably reach the existing side with a probability of 1. The same principle applies when two groups of living organisms occupy the same ecological niche with limited resources. It's inevitable that one group will eventually outcompete the other. Assuming equal conditions, the chance for each group to take over the entire niche is proportional to its initial population size. The same thing happens with different versions of a single gene. Even if they are initially equivalent, genetic drift will cause one variant to become fixed while the others disappear. The probability of fixation for each variant is equal to its initial proportion in the population. In a population of asexual organisms, only the descendants of a single individual will ultimately remain. I hope you've grasped the core idea. If all variants are equivalent, the chance for each one to take over the entire niche is proportional to its initial share. The displacement of all others and the fixation of just one variant will inevitably occur. I will be using a population of 48 women and 48 men. We'll start the model. I'm only keeping the lines that reach the final generation. The number of these lines will decrease until only one remains. This is a trivial fact, and it's what we call a random walk. And there we have Y-chromosomal Adam, who is the most recent common male ancestor for all men in the final generation. All men in the last generation have a Y chromosome from him. The Y chromosomes of all his other contemporaries have been lost. Then mitochondrial Eve appears. She is the closest direct female ancestor for all the women in the last generation. Note that other women lived at the same time as her, but their mitochondrial DNA didn't make it to the present day. I'll carefully redraw all the lineages, and what we get are called haplogroups. Haplogroups are groups of people who share a common ancestor through a specific line of inheritance. The concept of haplogroups is actively used by scientists to research the genetic past of humans. By comparing the DNA in the final generation, we can estimate when the haplogroups diverged. Knowing the time of haplogroup divergence and having statistics on their distribution in different regions, we can trace the migration of ancient humans. Based on the diagram we created, it looks like Adam is older than Eve. But notice that there is one haplogroup represented by only one person in the last generation. If it disappears, everything will change dramatically. Something similar happened in reality, but in the opposite direction. Adam was thought to be younger than Eve until a new haplogroup was discovered in 2012 in certain areas of Cameroon. This haplogroup is very different from all the others, and it significantly increased the estimated age of Adam. But let's return to our simulation. We'll take the next step, and that branch disappears. Now Adam becomes younger than Eve. As new branches disappear, the titles of Adam and Eve will also move forward in time. This means the titles of mitochondrial Eve and Y-chromosomal Adam are not static. Because men have a much wider variation in reproductive success, Adam is more likely to be closer to us in time, and this simulation confirms that. Sometimes Eve ends up younger, but on average, Adam is about 1.5 to 2 times closer to the last generation based on my model's conditions. To understand why this happens, let's look at an extreme example. If only one man leaves offspring, then the most recent common male ancestor becomes a representative of the previous generation. I hope this model has clarified who mitochondrial Eve and Y-chromosomal Adam are. I hope it's clear that other men and women lived during their time and for many centuries before them. This is a title that is passed on, so it's possible that someone watching this video will become the new Adam or Eve for humanity in the future. The only small condition is that a woman must have a minimum of 2 daughters, and a man must have a minimum of 2 sons. And what about our connection to all the other people who didn't make it onto the family trees we built? Let's take one person from the last generation. In the previous generation, they had two ancestors, their mother and father. Before that, there were four ancestors, two grandmothers and two grandfathers. In each subsequent generation, the number of ancestors doubles, assuming there's no inbreeding. In a small population of 96 individuals, which I'm using in this model, almost everyone can be an ancestor in just 7 generations. Now, let's look at the ancestors for other people in the last generation. Notice that almost everyone in the past has the same ancestors. I've highlighted in red all the people who are ancestors for every single person in the final generation. Those in blue are ancestors for at least one person. Let's take a closer look at the later generations. We'll go back through the generations until we find someone who is an ancestor to every person in the last generation. And here it is after 7 generations, we've found 2 such people. They are the most recent common ancestors for every single person in the final generation. But we're not going to stop there. We'll keep going until all the people marked in blue disappear. And now we've reached this point, which is called the identical ancestors point. Starting from this point, anyone whose descendants survive to the final generation is an ancestor to every single person in the last generation. This means that, from this point forward, all people have a completely identical pool of ancestors. From mitochondrial Eve, you can trace a maternal lineage to any person in the last generation. From Y-chromosomal Adam, you can trace a paternal lineage to any man in the last generation. From any person marked in red on this diagram, you can trace a mixed lineage to any person in the last generation. Scientists estimate that our most recent common ancestor lived between 2 and 5,000 years ago, while the identical ancestors point begins between 5 and 15,000 years ago. This is what it looks like over 960 generations. As a reminder, the people highlighted in red are ancestors to every single person in the final generation. If you go back several thousand years, almost everyone who lived then is our ancestor, and you can build a genealogical tree from them to you and to any other person. Now let's explore what we actually inherit from them. In this model, in addition to the Y chromosome and mitochondrial DNA, I also added 8 genes that we can track. Since we have a diploid set, there are 2 versions of each gene. I added a tag corresponding to that person's number to all the genes in each person. So, we have 96 versions of each gene. This will let us see what happened to the genes of each of the 96 original people over the generations. When a sex cell is created, crossing over occurs. A single set of genes is formed, and for each gene, one of the 2 versions is chosen. The sex cells from 2 individuals then combine, forming a complete diploid set of genes. Half of these genes come from the mother and half from the father. So, in the first generation, the offspring receives exactly half of the genes with tag number 1. When a sex cell is formed in the next generation, it will contain, on average, about a quarter of the genes with tag number 1. But since crossing over is random, the number could be more or less. After several generations, an offspring might not have a single gene with tag number 1 from their original ancestor. Even if crossing over were perfectly precise and an organism passed down exactly half of the genes from both the mother and father, given the size of our DNA, after 33 generations, there would be nothing left to divide, the offspring would receive less than 1 nucleotide. This doesn't mean you inherited nothing from an ancestor who lived dozens of generations ago. It's all random. The exceptions are the Y chromosome, which doesn't undergo crossing over, and mitochondrial DNA. It's time to see what happened to the genes in our simulation. This is the first person from the original generation. The black dots show when their genes can be found in the population over 960 generations. Do you see those dots? There. This person's 8th gene was the most successful, and that gene variant could be found for 9 generations. Starting from the 10th generation, there were no genes left from this person in the population. The second person from the original generation was more successful. Their 5th gene variant can be found in the population for almost 200 generations. The third person's genome disappeared from the population after 20 generations. Traces of the next 2 people can be found in the population's genome for almost 50 generations. You'll see a similar pattern for almost every person. And finally, with the 6th person, we see that their variant of the first gene has displaced all other variants from the population. After a few hundred generations, every person in the population has only this single variant of the first gene. The exact same thing happened with all the other genes. Initially, there were 96 variants of each gene, but within a few hundred generations, only one variant of each gene remained, having outcompete all the others. Let's take a closer look at the spread of the winning variant of the first gene. The dark color marks the carriers of this variant during the first 180 generations. Let's keep only those who, over the generations, were able to pass this gene on to representatives of the last generation, and let's connect the lines to show the gene's transmission path. It doesn't look as neat since the lineage alternates between men and women, but all the lines converge to a single person. I've marked where this happens with a green bar. Let's move forward in time a bit. Now, one of the branches will be lost, and the title of the last person from whom everyone received this gene variant will move forward. So, exactly as with Adam and Eve, we can identify the most recent common ancestor for any gene or section of DNA that has not undergone crossing over. The people marked in red are the common ancestors for all people in the last generation over 960 generations. The bottom diagram shows the 2 chains through which mitochondrial DNA and the Y-chromosome were passed down from the depths of time. Only after Adam and Eve did they branch out to reach everyone in the last generation. There are similar chains for each of the 8 genes as well. Even though most people in the past are our ancestors, only those marked with black dots can truly say that something was passed down through them to the last generation. In the final generation, all the inherited material we tracked in this model came from just 10 people from the first generation. If we don't account for mutations, everyone should eventually become clones with a completely identical set of DNA. We didn't become clones because mutations exist. It is mutations that create the basis for diversity. Thanks to mutations, we can distinguish different haplogroups from one another, as each separate branch acquires its own unique mutations, which set it apart. By comparing them, we can build a phylogenetic tree and calculate the time of divergence for different branches. But more on that in the next video about molecular clocks. I covered in detail what would happen without mutations in my video about the advantages of sexual reproduction. In asexual reproduction, the offspring of the most successful or best adapted organism will displace all others. In sexual reproduction, it is the genes that compete with other variants of the same gene. That's why many people view evolution from the perspective of genes competing for survival and replication, rather than organisms or species. Organisms are merely the environment where the competition between different gene variants takes place. Let's look at the history of a single gene from the perspective that genes are the main agents of evolution. A gene's lineage is a long chain, one person thick, stretching back into the past. Only at the end of this chain are there many branches that, using a population of people, test different variants. Most of these branches will disappear forever, and only the most successful variant will determine the direction and become the continuation of the long chain stretching through the ages. That's all for today. Like, subscribe, and leave a comment. A special thank you to everyone who supports me on Patreon. Bye everyone.